Welcome to Biology at Priyajit, my dear viewers. Today, have you ever wondered how vaccines are developed safely in the biotechnology laboratories, or how living cells are preserved for years and used again for research and medicine? All of this is possible through advanced animal cell culture techniques. In today's class, we will explore culture techniques and applications, including types of animal cell cultures, the scientific principles of cryopreservation, and the crucial role of animal cell culture in vaccine development. So, these techniques form the backbone of modern biotechnology, medical research, and vaccine production. So, what is animal cell culture? Animal cell culture is basically refers to in vitro maintenance and growth of animal cells, tissues or organs under controlled laboratory conditions using suitable culture media and sterile environment. Types of animal cell culture. Animal cell cultures are classified based on origin, growth characteristics, and morphology. Based on origin or primary culture, it is derived directly from animal tissue, and the cells obtained by mechanical disaggregation, enzymatic digestion such as trypsin digestion. It contains heterogeneous cell population and it shows finite lifespan. So what are the characteristics of primary culture? It is closely resembled to original tissue, and there are limited cell division. It is highly sensitive to contamination. Examples: primary fibroblast culture, primary kidney cell culture, etc. This is the image showing the different types of animal cell culture, different aspects of animal cell culture. Now we'll go for secondary culture after discussion about primary cell culture. Secondary culture is obtained by subculturing the primary culture, and cells are transferred into fresh culture media for further growth. A cell line is a population of cells capable of repeated subculture. So what is cell line? Cell line is a population of cells capable of repeated subculture. Types of cell lines: finite cell lines and continuous cell line. Limited divisions, that is about 20 to 80 populations doubling and undergo cellular senescence. Continuous cell line, that are transformed or immortal cells, they divide indefinitely. Examples: HeLa cells, etc. Based on growth characteristics, they can be anchorage dependent, which require solid surface attachment, and it can grow as a monolayer. And anchorage independent cells grow freely in suspension. Examples: blood cells. So based on morphology, cells may appear as fibroblast-like, it can be elongated, epithelial-like, which can be polygonal, and lymphoblast-like, which is spherical in nature. Culture media. We know that culture media usually contains amino acids, vitamins, glucose, inorganic salts, fetal bovine serum, antibiotics, etc. So types of media: it can be natural media like serum and plasma, and artificial media can be MEM, DMEM, or RPMI 1640. Cryopreservation. Cryopreservation is the long-term preservation of cells at ultra-low temperature. That is nearly about minus 180 degrees Celsius using liquid nitrogen. Principle: low temperature always stops metabolic activity, and cryoprotectants always prevent ice crystal damage. So what are the cryoprotective agents? Such as DMSO, that is dimethyl sulfoxide, and glycerol. Steps of cryopreservation: it harvests healthy cells, it add cryoprotectant, slow cooling, that is approximately 1 degree Celsius per minute, store in liquid nitrogen, that is in about minus 196 degrees Celsius, rapid thawing at 37 degrees Celsius, and remove DMSO. These are the steps of cryopreservation. What are the advantages? You can use it for long-term storage. It maintains genetic stability. It is useful for cellular banks. Application: stem cell preservation, IVF embryo storage, in vitro fertilization embryo storage, and conservation of endangered species. Research: cell bank. Animal cell culture in vaccine development. Importance: viruses require living cells for replication. Therefore, animal cell culture plays a major role in viral vaccine production. What are the types of cell cultures used? Primary cell culture. Examples: monkey kidney cells, diploid cell strains, human diploid fibroblast. For examples, you can say WI38, and continuous cell lines. Examples: Vero cells, MDCK cells, etc. So what are the steps in vaccine production? First of all, you have to select suitable cell line, then you have to go for virus inoculation, then virus replication, then harvesting the virus, inactivation or attenuation of the virus, then purification, and lastly formulation and quality control. Types of vaccines product produce: live attenuated vaccines, inactivated vaccines, and recombinant vaccines. Examples of vaccines: it can be polio vaccine, rabies vaccine, influenza vaccine, COVID-19 vaccines, etc. Advantages are following: it is safer than animal-based methods, large-scale production is possible here, and reduced contamination risk, ethical alternative to animal use. But there are some limitations such as expensive laboratory setup, requires strict sterile conditions, and risk of contamination, genetic instability in continuous cell lines. So in today's class we have learned about the types of animal cell cultures, the importance of cryopreservation, and the vital method of animal cell culture in vaccine development. So these techniques are essential tools in modern biotechnology, medicine, and safe vaccine production. If you found this video helpful, don't forget to like, share, and subscribe to this channel. Keep studying, keep growing, and always remember, biology and biotechnology is beautiful. Thank you.